Hello and welcome. This is Get Ahead Institute. In this video, we are going to talk about cellular communication and messengers. Okay, so we are going to briefly explore what these cell to cell adhesion junctions are, and then we're going to talk about intercellular communication as well, how these cells communicate. Then we're also going to talk about the messengers that are involved in this communication. Okay, and then the mechanisms which these cellular messengers use uh, to communicate. Okay, for cell-to-cell communication, how do these messengers come into play? Then, of the mechanisms we're going to talk about, we're going to explore deeply the G-protein activation pathway for cellular messengers. So let's get right into it. So you know that cells being the individual units that make up the organisms that we all see have to communicate one way or another. So some of these cells actually have to function as units, as you're able to see it in some tissues and some organs as well. So that level of communication, that synchronicity, so to say, has to be built up. The cells need to communicate and act as one. So how do they achieve this? These cells are adhered together, as you see in tissues, and these cells are also able to communicate with each other and so that they can act in a sequential manner, in a sequential or in synchronicity. That's what we're going to explore in this video. So I'm going to go ahead with cell-to-cell adhesion. In cell-to-cell adhesion, there are different cell-to-cell connections that we have. These are cell-to-cell connections. Adhesion, we're connecting the cells. Okay, and these cells can either connect with each other through either gap junctions or tight junctions. So from the word gap junctions, these are junctions which allow cell to cell transfer of material without utilizing the ECF route. Okay, that means the cells are able to communicate directly, they're able to exchange materials directly without having to pass through the extracellular matrix, without the materials having to go out of the cell, then into the other cell, but they can go directly from this cell to the next cell. Okay, and then these gap junctions are also called connexons. These connexons are made of six subunits known as connexins. Okay, so the individual subunits of a connexons are called connexins, and there are six of them in a connexon. Then, when it comes to tight junctions, on the other hand, these junctions bind cells tightly and do not allow any material exchange between the two cells. Okay, so let's explore further these tight junctions. We have desmosomes and hemidesmosomes. So these are cellular junctions that connect adjacent cells in tissues, providing the strength and stability by anchoring intermediate filaments within the cells. Okay, so you're going to have an arrangement which is sometimes described as a spot weld. Okay, where you are going to have two adjacent cell membranes, different cells, and you're going to have a protein here, the protein here, attached to intermediate filaments like that. So it looks like a spot weld. Okay, spot weld. Those who have seen people who are welding and stuff, it looks like a spot weld. You just weld something on that spot. All right, so that is about tight junctions, and these are desmosomes and hemidesmosomes. So what's the difference between the two? The desmosomes, these ones will be found on the adjacent membranes of the cells. Okay, and then the hemidesmosomes will be similar, but hemi coming from the word hemi means half, so it's half a desmosome, and these ones they anchor the epithelial cells to the basement membrane, helping to stabilize and support the tissue structure. Okay, so as we'll see in the next picture, and then proteins that are involved in these connections are occludins and claudins as well. Okay, so this is a picture showing your desmosomes and hemidesmosomes. So when you have your epithelium like that, we're saying that the desmosomes will connect the cells here where the cells are adjacent, and then on the basement membrane here you have your hemidesmosomes. So it's half a desmosome in the sense that you have one spot word there with the intermediate filaments. Whereas if it's on the adjacent cells, you're going to have one protein there, another protein on this other cell, and intermediate filaments like that. That's your desmosomes and hemidesmosomes. Okay, so the proteins that are involved, like I said, you have some claudins there and occludin there. That's just a visual representation of how these cells help, how these proteins help in these cell to cell adhesions. All right, then this is another picture showing you your tight junctions and your desmosome. Like I said, it looks like a spot rod there with your intermediate filaments coming out like that, and then your tight junctions. These are occluding junctions, which do not allow any material movement between the two cells. All right, so we move on to cellular communications. These cells, as much as they are tightly bound together, they have to communicate one way or the other. So let's explore how these cells communicate. So there are various ways and various names given to a type of communication depending on where the cell is and where the target cell is. So for endocrine, we are saying that it refers to the type of signaling where the molecules released by endocrine glands into the bloodstream to distant target cells. So the target cells in this case are very distant, and they utilize the bloodstream to send these hormones and chemical messages to their target cells. Okay, and this is endocrine kind of communication. You don't have glands with this kind of. You don't have. I mean, the glands don't have ducts, so they have to release their materials into the bloodstream. And for autocrine, we have a signaling where a cell releases substances that will affect its own activity. So the cell itself, we release that material that will affect itself. Okay, that is what happens in autocrine communication. And in justacrine, on the other hand, from the word juxtaposition, that means next to. So signaling between adjacent cells that are right next to each other. These require direct cell-to-cell contact. Okay, let's say for example, macrophage and um, macrophage and the phagocyte, where a macrophage acts as an antigen presenting cell that presents the antigen to the phagocyte for this what is it called, for phagocytosis. Yeah, so during that interaction the cells have to come together and they have to meet up, and that cell-to-cell contact has to happen in just a justacrine communication. Then in paracrine, on the other end, we have signaling where a cell releases signaling molecules to nearby cells in the same tissue. So the cells are just nearby or at some distance, not very far away though, but in the same tissue. Okay, but on neurocrine or the neural communication rather, it refers to the transmission of signals between neurons in the nervous system and including synaptic and non-synaptic communication. So we can include the neuromuscular junction here as well as part of neural communication. Okay, so as these cells are communicating, we have these things we have to know of, which are known as receptors and messengers. So a receptor is a protein molecule that responds to binding of a ligand or a messenger. So this receptor is more like this is a place where the molecule that you're sending to, the molecule you're using for this communication is the one, the messenger we're speaking about, or a ligand is the one that's going to bind to a particle known as a receptor. And these are protein in nature and they are transmembrane. Some of them are inside the cytosol, but you have various types of them, and some of them have enzymic activity, like you can see in the diagram. Some of them have intrinsic enzymic activity. Some of them are ion channels. So the receptor itself is also an ion channel where when it binds, the pore opens. Some of them are linked to an ion channel. Some of them just facilitate opening of an ion channel. Those are the different types of receptors that you can have on these cells. Then, when it comes to messengers themselves, messengers in cellular communication are the molecules that transmit signals. They're the ones that we use to transmit signals from one cell to another, influencing the various cellular processes. Okay, they will bind to the receptors that we talked about, and there are two types of messengers mainly. We have a first messenger and the second messenger. What is the difference between the two? So, the first and second messengers are, for the first messenger, this is a chemical messenger that comes from the outside of the cell, okay, or the ECF, and activates a receptor. Most of these receptors are on the membrane. Okay, most of them receptors for the first messengers are on the cell membrane. Okay, so the cell membrane, like that, is the nuclei, cytoplasm. Then on the cell membrane there, that's why you have your receptor for the first messengers. Okay, then for the second messenger, the criteria is that it comes into the cell. Okay, it comes into the cell. All they have to be produced inside the cell due to activation of a receptor. So first, the receptor has to be activated by a first messenger, by binding of a first messenger. Then the second messenger can be produced either by, we said that some of the receptors are ion channels or influence opening of ion channels or other types of channels. So then it can come into the cell due to opening of that channel by binding of a receptor that we said the first messenger has bound, and then uh, it influences opening of a certain channel that will allow the second messenger to go into the cell, or the second messenger will produce right inside the cell there to bind wherever it needs to bind. So an example of one that comes from outside to inside the cell is the CA2 binding during muscle contraction. It's the second messenger that gets into the cell after you have activation of DHP receptors, the dihydroxypyridine receptors. Once they bind, and once their voltage getting to channels for calcium, by the way. So once they sense a change in voltage brought about due to the action potential, they're going to open, and then the second messenger. So the first messenger in that case is uh, your membrane potential, which I talked about in neurocommunication. Okay, neurocommunication, the action potential, and the like. So then the second messenger is produced. Okay, it's released, which is the calcium, and it goes into the inside of a cell, and it's going to cause muscle contraction one way or the other. Okay, and then we are saying there are some that are produced inside the cell. An example, we have cyclic AMP, which is very common, cyclic GMP, inositol triphosphate, and diacylglycerol or DAG, and calcium 2 plus as well. These ones that can be produced inside the cell, like for example, CA2 in this case is the one that is coming from the sarcoplasmic reticulum inside the muscle cell. Then for diacylglycerol and cyclic AMP, these ones have to be produced inside the cell by activating of certain enzyme, certain cyclase enzyme. Then inositol triphosphate as well. When you talk about the muscle, these ones will also be produced so that the inositol triphosphate will also be produced so that it can cause further opening of more calcium channels. Okay. Then types of second messengers, we have hydrophobic, hydrophilic, and gaseous second messenger. Then those ones which are hydrophobic, we know that they don't mix with water, so they are membrane associated. We know that the lipid bilayer is made of lipids, so their receptors are there so that they can easily communicate in that manner. If their receptors were inside here, because they don't mix with a hydro a watery environment, because they're hydrophobic, it will be hard for them to be bound to bind to their receptors. Okay, so these are membrane associated. And these ones, the hydrophilic ones, are located in the cytosol. That's where most of the action happens, because this is where the water is in the cytosol. You have a lot of water there. And then for gases, these diffuse through the cell membrane. An example is nitric oxide. Yeah, these ones are able to diffuse through the cell membrane and to the cytosol, and they're able to attach to receptors that are on the nucleus themselves. Okay, so let's start on second messenger. So these second messengers, what is their function? Second, first messenger, what is their function really? So In overall, their function is to amplify the signals. Okay, the signals that are sent, we want them to amplify because uh, the body is very economical, so to say, in terms of sending a few packets that have to be received somewhere. Let's say, for example, you have certain molecules in the body that are scarce. Let's say, for example, some neurotransmitters that are very scarce, like glutamate, they are amino acids that have to be constantly replaced and recycled. So, to make sure that you are economic and utilize little uh, messengers, but then you have to amplify the signal with those molecules that can be easily easily manufactured in the cell and are readily available, those that are not toxic and the like, which their hyperproduction is not toxic. So you have to amplify the signal so that you maintain those you maintain high integrity of the information being transferred across these cells with using minimal resources for those that are minimum, for the resources that are minimum, for those signals, for those messages that are minimum, so to say. And another function is to connect the cell surface to the nucleus. What do we mean here? Okay, so what we mean here is that there's some molecules. Okay, the cell has to communicate through through various means. Okay, like we said, the cell nucleus is the one that is responsible for maintaining the action of the cell, so to say. So, how does this nuclei, com- or let's say, control things that are happening on the cell membrane? So, receptors can be used, and messengers can be used either from the nucleus to the cell membrane or and the other way around. So, this is how we connect the cell surface to the nucleus. Okay, and then finally, last but not least, or not the only ones we have modify the function of the enzyme, the functionality of the enzyme. We need to modify some enzymes that are occurring inside some enzymic activity that is occurring inside these cells. So how do we do that? We're going to use these messengers so that we can either inhibit or promote uh, enzymic activity that is happening inside these cells. All right, those are the functions. And there are four mechanisms by which these chemical messengers will act. Okay, firstly, we have ion channel activation. So, for example, inositol triphosphate causes CA2 channel opening in the sarcoplasmic reticulum, like I had said. So, they can cause ion channel activation or activation of enzymes and modification of the activity, like I had mentioned earlier, within the cell. So let's, for example, CA2 binds to comodulin in smooth muscles. What does that cause? It causes the activation of what is known as the comodulin dependent light chain kinase. So that will activate that enzyme so that muscle contraction can occur in the smooth muscles. Then we have also direct activation of DNA transcription so that we can transcribe certain proteins that are needed in the body. Then another one is G-protein activation. G-protein activation, we have G-coupled receptors that activate certain proteins so that they can achieve a certain function in the body, as we will see. So for G-protein coupled receptors, we're going to explore deeper into them just a little bit here. So for G-protein uh, coupled receptor complex, it looks something like this. You're able to see here. So what this complex is all about, we have a receptor here that is coupled to a G protein, and this G protein has to be activated so that it can influence a certain activity on an effector, so that this effector can produce a second messenger. Like we said, some second messengers are produced inside the cell. So we have a receptor that binds the ligand. Okay, and then after binding on that ligand, it activates your G protein, and that G protein is going to activate an effector that is going to produce your second message. So your effector is going to produce your second message. That's what we are saying. All right. So how does this whole activity happen? We have this diagrammatic representation here that I'm going to briefly elaborate. Okay, so your extracellular or first messenger is going to bind here into the G protein coupled receptor. This is receptor side of it. Once it binds here, it's going to activate uh, this receptor. what is the action of this receptor? It's going to cause this G-protein intermediary. So what we mean by G-protein coupled receptor, we mean that this receptor is coupled to a G-protein. That means it's connected to a G-protein one way or the other. So they are coupled. That means they are connected together. So activation of this receptor is going to also activate the G protein. And what does the activation of the G protein result into? The alpha subunit. It has the G protein has three subunits, by the way. Alpha, we have the alpha, the gamma, and the beta, as you can see there. So these ones, once you activate it, the alpha subunit is going to move, okay, and it will go to activate adenine cyclase, which is an effector protein that we talked about. So that effector protein is responsible for changing that receptor protein is responsible for changing ATP into cyclic AMP. Why do we need cyclic AMP? That cyclic AMP is our second messenger. We need it as a second messenger uh, to do a number of things. Okay, we need it to activate certain proteins and also to do other stuff. There are other stuff that they can do, but here we're going to focus on activating a protein kinase. Okay, a protein kinase is an enzyme that phosphorylates proteins. And then phosphorylation of a protein, adding a phosphate to it will activate that protein. So how does that come about? Like we had said, firstly, the alpha subunit is going to detach from the G protein after it has been activated by this receptor by binding over the extracellular first messenger. Then once the alpha subunit goes to the adenyl cyclase is going to change ATP into cyclic AMP. Once it changes that, it's going to cause release of two inorganic phosphates as well. Nothing to mind there. But then this cyclic AMP is the one now that is going to act as a second messenger and it's going to do a number of functions in the cells. So for example, we have activation of a protein kinase A. So protein kinase A, the function is to activate a protein. Okay, so this is initially inactive. It is activated by CMP. So protein kinase A is activated by the CMP that is produced as a second messenger. This CMP, this is going to be activated, the protein kinase A, and then due to the activation is going to require energy in form of ATP and ADP there, so that this phosphate can be used to activate this protein kinase. So you remember that enzymes are also uh, proteins. So for them to be activated, you have to phosphorylate them. That's where we are putting this gamma phosphate there. So after that has been activated, we now have the action of this protein kinase. What is the action of this protein kinase? It activates a protein, it phosphorylates. Every enzyme that you see that has the word kinase, it is adding a protein, rather, it's adding a phosphate. It's involved in movement of a phosphate. Okay, so in this case, we are utilizing uh, the phosphate that we get from ATP, and then we are activating a certain protein by phosphorylating it as well. Like we had said, for us to activate the protein kinase, we have to phosphorylate it. Then for this protein kinase, once it's activated, for it to activate another protein, it has to phosphorylate it as well. That's the function, to phosphorylate it. So we are getting an inactive designated protein and activating it here. So that these proteins, you know that they come in various forms. This protein can either be another enzyme, it can be a protein that needs to be activated for particular reasons. So that is what is going to be happening in your, and then that is going to elicit a certain cellular response that we are going to see uh, as the ultimate goal of this G protein activation. So that is that on G protein coupled receptors, and that comes to the end of this video on cellular communication and messengers.